So, welcome everyone. I'll now turn it over to David and Ratan to um, uh, introduce the entire group and get the discussion going.
Okay, I'll just start off by saying that, yeah, been leading this Pennycress thing on my side of the coin for the last few years. And Ratan came in, I think, maybe 3.5 years ago. And he's really pretty much just taken everything over, and he's left me a little bit to do. And so what he's going to talk about today is largely a lot of stuff that he's been up to in promoting Pennycress, and really has been pushing the program. And so with that, I'll turn it over to Ratan.
Thanks, David. So I'm Ratan Chopra. I'm a researcher here at the University of Minnesota working with David Marks. And today I'm going to present about what we did over the past year for Pennycress trait discovery, and the work is being presented on behalf of the group. I mentioned some names on the slide here. I'm going to present some of the work Matthew Ott performed over the last year. He's going to talk on a slide, and Christian Rai, and among other people. And before I talk more about Pennycress, I just want you to see this busy slide where you can see there's a bunch of institutions and funding agencies that have supported this Pennycress program, and the list is growing year by year. Like we have the CAP grant, which is I Prefer. Recently, we were funded by DOE, which has been proposed to be called as DOE iPrep. And the funding has been coming from various other sources. And so it's a team effort. With that, what I plan to present today is 4 things. One, status of integration of crucial domestication traits that have been identified in the program. And then I'll cover a little bit about trait discovery, how we are trying to solve some of the traits that we don't have a solution for. And then what we are doing to keep the program robust by developing mutant resource that would stabilize the program for the long term, and few announcements that happened over the last 12 months in regard to the Pennycress program. So with that, just a brief introduction about Pennycress. Pennycress belongs to Brassicaceae family. It's native to Eurasia. It's been naturalized throughout Northern America, and more importantly, it's very closely related to model plant Arabidopsis. And why this is important, because back when Kevin Don and David Marks started this work of Pennycress genomics, one of the things they were able to identify was there was a close relationship between Arabidopsis and Pennycress. And the relation was to this extent that even the gene orientation was very much conserved between these 2 species. And because of that, there was promise that we could use Arabidopsis research translate that information to Pennycress and develop solutions for the traits or improve Pennycress in a quick manner. And to do that, in using that approach, we've already kind of solved many crucial traits that are important in Pennycress. For example, like wild type Pennycress has a lot of shattering issue. We identified a line that does not shatter. And what we were able to do is not only just identify the line, but go identify a specific gene, what the mutations are. That allows us to develop molecular markers that could be used in the breeding program. Similarly, wild type Pennycress has a lot of glucosinolates in the form of synegrin. And glucosinolates is present in more than 200 different forms, but the type of glucosinolate that is present in Pennycress is not desirable for the meal when you try to feed it to animals or poultry and things like that. And we found a line that had a huge reduction in glucosinolates, and we were able to identify the mutation for it and characterize the gene to some degree. At the same time, wild type Pennycress has a lot of uric acid, which is desirable for biofuel industry, but at the same time, uh, having high oleic acid would be useful for biodiesel or application in the food industry. So what we've been able to do is identify these traits, identify genes associated with this, and not only genes associated with it, but also specific mutation for which we can design molecular markers for, and then use that in our breeding programs to introduce these traits. So with that, I'm sorry. With that, so using those traits, what we've been successful over last 3 to 4 years now is we have the molecular markers. We can stack these into a single 106 background and see how they perform out in the field conditions. So already we have lines like high oleic reduced shatter that basically has 60% oleic acid and does not shatter in the field conditions. We obtained about 17 pounds of seeds this summer, and those seeds have been provided to Dr. Wells for increasing. That would help with the agronomic trials in the near future with the lines having 106 background, but some of the improved traits. Similarly, we have high oleic seed that was increased on St. Paul campus this year, uh, for which we obtained over 200 pounds of seed that was currently present at AURI. And Connie has been working with AURI and the other collaborators to supply that oil and meal as necessary to the groups and kind of understand the application and develop products around this particular line. At the same time, we made more progress. This fall, we planted some couple of quadruple mutants. Both of them have zero uracil acid and zero glucosinolates, but one of them consists early flowering 6 and the other one consists rod 1. But this picture was taken last week. As you can see that the establishment is nearly as good as 106. It's not uniform, and that is due to that we didn't have rain after we planted it, but still we can see that these quadruple mutants or triple mutants are germinating. They look good, at least until now. We don't have a ground snow cover yet, so we don't know how that's going to affect the later in the year or later in the season. But what I want to say is we've made good progress in terms of stacking these traits that I discussed. And at the same time, more cleaning is going on. Like David Marks has led the efforts to kind of keep cleaning these quadruple and pentamutants to create as pure of a line with these improved traits as possible. So we are anticipating next year we'll have like pentam lines with at least 5 different genes with a much cleaner background of 106 that would be going to field. So that's with trait integration where we stand. Now I'm going to talk a little bit more about trait discovery. So we've identified solution to like low glucosinolate, improved oil properties, reduce shatter, early flowering. But to make pennycress commercially viable, one of the things that is needed is you want high oil content, high protein content. And these are quantitative traits, and it's not been as simple that you can go and find a gene that would control this trait. Yes, we could do that, but it's not been such an easy trait to solve. So to identify some of the high oil lines, what we've done over last 3 years is selected lines based on NIR predictions for high oil content and try to look at their heritability year after year. And what we found was transparent testa line, which is yellow seeded pennycress lines, always had higher oil content compared to the corresponding grown wild type. Um, and you can see that there is year to year variability in the total oil of a given line, and it's highly dependent on the year that was grown in. But it's important to note that transparent testa lines always had a higher oil content. But we did have one line, E5248, that had much about 5% higher oil content compared to the corresponding wild type for a couple of years. And we are interested in learning what is causing high oil content in that particular line. Similarly, another quantitative trait that we want to increase protein content. Again, increasing protein content is of a value because uh, there would be a lot of meal that would be left over after we crush the pennycress seeds. And there is a growing interest for plant-based protein in the industries, and people are moving towards this approach. And we have a good collaboration on campus with the Plant Protein Innovation Center. So one of the approaches we have taken for increasing protein content in pennycress is, again, look at the NIR predictions. So our average protein content for the year would be around 24, 25%. But then we do have lines that have higher protein content and some lower protein content. And we've done some validation using combustion assays to see if these predictions are true. And indeed, we've seen some strong correlation. And once we identified these lines that have consistently shown higher protein content compared to the wild type, we've run them on the gel. We haven't seen huge differences in the band patterns. But what we have seen is when we did amino acid analysis for these lines, we've seen some differences, and we are investigating further. And this work has been mostly carried out by Krishan Rai, who was a part of the team during this year. The other trait, even though I mentioned that we solved it now, reducing glucosinolates in pennycress, but there are certain challenges that we have seen whenever we reduce glucosinolate throughout the plant. We see that there is some sort of insect pressure that could damage the plants, and we are working towards understanding this. Like we've seen lines that have no glucosinolate, intermediate glucosinolate, and for some of these, we've identified genes successfully, and some of them we are still working on. What's important, what we noticed was plant height did reduce with the reduction of glucosinolate, at least in these three lines, and this one did not. And we are further investigating whether this would impact the total yields overall when you reduce the glucosinolates in pennycress. And then since we are concerned about reducing glucosinolate throughout the pennycress plant, one of the things that we are focusing on is working with Bob Cook in entomology, Catherine Bushley, Cody, trying to understand how these reduced glucosinolates would interact with different pests or nematodes and what their relation would look like. Even though we have nutty, but we are trying to work towards characterizing these other glucosinolate lines, find those genes so that we can have reduction only in the seed specific region or somewhat reduction in pest pressure on the plants. The other trait that we've all been concerned about pennycress is seed dormancy. As we know, pennycress is a weed, and it does carry this dormancy issue naturally, and seed dormancy is a very complex process. But what I'm showing here is just how do we reduce fresh seed dormancy, and that was something that we surveyed this year first time. After our plots were mature, we had over 250 plots on St. Paul campus. We generally plant in 4x4 plot size, and this year we decided to leave the plants after harvest was done for single plants, and there were a bunch of plants remaining on those plots. We went after 2 weeks to look at how these plots look. Do they have any pennycress plants growing in them, or if they are, how robust do they look? And then I gave a score of like 1 if there was nothing growing, 3 the plants had been established, but 5 when they were really vigorous. And using those using that scale, what I was able to do is basically see that wild type 106 did not necessarily have huge fresh seed dormancy, but there were more other lines that just were robust, had a very nice rosette. And when we started investigating further into what these lines are, what we found was all of these lines were transparent testa, which was yellow-seeded pennycress lines. So that made us believe that, well, if we use yellow-seeded pennycress, maybe we'll have higher oil, higher protein, and don't have to deal with the fresh seed dormancy issue in pennycress. So because of these encouraging results, one of the things Matthew Ott did for his thesis to understand how these TTs are different compared to wild type and what makes them different. So he's going to talk a little bit about transparent testa mutants, and then I'll take it back over. Matthew, go ahead.
Thank you, Ratan. And I'll just start by talking about the data in the figure with the colored bars, and I'll describe the experiment a little bit. This data comes from a field experiment conducted in St. Paul. We used a cone seeder to plant 100 seeds per plot of a given genotype and seed treatment, and there were 4 replications over 3 seasons. So to orient you more to the figure specifically, there were 2 genotypes that we're showing. We've got wild type, which is represented by the light colored bars, and TT2, transparent testa 2, which is represented by the darker colored bars. And we had 2 seed treatments. There was an 18-hour soak in plain sterile water and an 18-hour soak in GA3, followed by a dry down before planting. So just use desk fans to dry the seed back down. And then the orange bars represent an untreated control. And that actually, the untreated control is most relevant for the dormancy question in Pennycress, because if we're going to be going through and harvesting with the combine, and inevitably some of that's going to be a leaky harvest, that seed won't be treated, and that is a potential weediness problem. We see in our breeding fields year after year, despite not having used them for several years, Pennycress keeps coming back and keeps coming back. So, I mean, it is a weed, and dormancy is a major driver behind that weediness. So looking at how transparent testa does in an untreated seed, I guess, treatment, the untreated control, it outperformed the wild type Min 106 in all 3 seasons. F19 represents fall 2019, S20 is spring 2020, and F20 is fall 2020. And in all 3 seasons, transparent testa 2 outperformed wild type Min 106. In the first season, fall 2019, we planted it a little deep. It was 3/4 inch. So that's why a little bit less emergence overall, but still TT2 did better. And the spring 2020 and fall 2020 planting depths were more of typically what we would do, out 1/4 inch for spring 2020 and 3/8 inch for fall 2020. The soil conditions were a little too fluffy in fall 2019, went a little too deep. But that being said, we are seeing emergence rates of around 90%. And that's great news in terms of the issue of dormancy, at least in this soil type of St. Paul. And it means that we're less likely to have a weediness problem with transparent testa 2 genotype of Pennycress. And I think Covercress has done some research, and John Sedbrook has done some on other TT transparent testa mutants of Pennycress, and have seen similar results too. So yeah, that's exciting news. Less of a weediness problem in these soil conditions. However, in the figure with the blue bars, we kind of see the opposite trend. And this is in sandy soil. Now, this was also done in the field in St. Paul, but I collected soil from Becker, Minnesota. And that is classified as just a like basically pure sand soil. It's not even loamy sand. It's just sand, pretty much. So, and we found that TT2 did not germinate well in sand. and this is under rain-fed conditions. So the likely mechanism behind this is just water, whether it's imbibing. We're still trying to dig into the actual mechanism of what's going on, but we do know, and also we find in the Arabidopsis literature as well, that transparent testa genotypes have lower rates of germination in water stress conditions. So is this going to be a major problem? It's to be determined. Looking at a map of the Midwest recently, I found that this classification of soil, this sandy soil that we found in Becker, is on very few acres relative to even a loamy sand in Minnesota. It's like 1/100th of the acres compared to the loamy sand. So next steps would be interesting to try how transparent testa 2 does in loamy sand, because that represents more acres in central Minnesota than just the pure sand soil.
Matthew,
So yeah.
it's Mark Messmer. So my just throw this theory out. I don't know how closely you actually are able to look at the actual seeds, but my theory is, and I think we've seen this, and one of my concerns, and one of the things we have to pay attention to from a covercress standpoint, commercialization, is that I actually think what happens with the transparent testa genotypes is that you actually do get germination, and you get it germinates and dries out and dies.
Yeah,
In a situation
so
like that.
that is something that
I think you get really fast germination with those things. And one of the concerns I have is that we just have to continue to look at, and we've seen some of it, having had the drought that we had this fall down here. I would throw that out as a theory.
Sure, no, that's definitely something that we're trying to dig into as well. And this is a bit of an ongoing experiment. I left these boxes out in the field to see if, getting more precipitation over the winter, see if we see any germination at any point. But no, that's a valid theory. And
the theory, of course, would be that the black seed doesn't, the wild type doesn't germinate as quickly, and it kind of waits until there's enough continued moisture to go ahead and go,
Right. Yeah. Well, at least in this experiment, we got about 1/4 inch of rain on the sand, and then there was basically nothing after that 1/4 inch of rain for several weeks. And with that amount of rain, the black seeded wild type did germinate at the expected rate.
Okay. Okay.
But the TT2 1/4 inch wasn't enough.
Okay.
So.
Well, it's just it's an interesting phenomenon that I think we have to figure out.
Yeah, absolutely. So, but yeah, it's something, I think trying more soil types, different loamy sand, I think would be interesting, or a sandy loam as well, because that represents actual more acres in the Midwest than just the pure sand in Becker. So definitely more experiments to be done and more things to be learned. And yeah, if there are any other questions, I can take them now or at the end, and otherwise I can turn it back over to Ratan too.
Okay, I can continue. So like, Matthew said that we can solve seed establishment or dormancy issue with transparent testa, but as you saw that there is possibly a challenge that we could incur over time using these transparent testa lines. We know that under optimal conditions, transparent testa can do very well, but under some sort of stress condition, it needs to be determined. But our initial studies have suggested that we could run into challenges. But it's not, even if we decide to proceed with transparent test our lines, seed germination and dormancy is a very complex trait. And there are several things we still need to understand about seed dormancy and germination in pennycress, because there's a lot of factors that can affect these traits. For example, like when the pennycress is maturing in the spring, do we have a cooler spring versus a warm spring? And how does that affect the dormancy of the seed? How does that affect the quality of the seeds we have? And there is some literature out there suggesting that when you have a warmer spring, you're going to have reduced seed dormancy, versus when you have cooler spring, you're going to have increased seed dormancy. That's one question we need to answer. The second question is, like, as Matthew pointed out to, and as Mark mentioned about this year's fall, that you could have poor germination when you have poor rainfalls and less rain events in the fall. And then in that case, can we bury the seeds deeper? And that's also important to learn, because until we solve all of these issues, or some of these issues, it's going to be an ongoing problem. But and this is represented by this figure here, as you can see that this is putative seed dormancy pathway, or you can look at seed germination pathway. There's multiple genes that are involved. For example, there is a gene called BZIP67. When you have heat treatment during maturation, you will have reduced expression of DOG1, resulting in reduced seed dormancy. When you have cooler temperature treatment, then you're going to have increased expression of ZIP67, resulting in increased seed dormancy. At the same time, on this side of the pathway, you see that there are a lot of genes that are involved in seed storage protein. That is also important for modifying our meal property. But at the same time, when we do that, we can also regulate the AB and GA level, and some of it's regulated by SOM. And SOM is dependent on light. For example, like you have a light condition and pennycress is dependent on light germination, you get rid of this particular gene, then you could have light independent germination. So using just these two targets, maybe can we solve some of those three issues that I mentioned? And I'll show you a couple of slides that we think we have a solution for three of those problems that I mentioned, and how we've characterized that. So we have mutations in BZIP67 and SOM. So for example, to test if pennycress lines can germinate under low water potential, to do this, we could obviously do it in the field conditions, but we wanted to test if this is going to work in the lab conditions first. To create those low water potential conditions, we use PEG 8000 and prepared 15% PEG, 20% PEG, and 25% PEG. And then put the seeds into Ziploc bags and look for germination every 24 hours. We had anywhere between 30 to 50 seeds in each Ziploc bag. And what we were looking for is what is the total germination among these lines that have like BZIP67, wild type, SOM, and TT2. And what we saw was mostly all of these lines that were potential candidates for reduced seed dormancy, or that can germinate good under water, reduced water potential or reduced light, germinated within 48 hours. And the way we classified germination is when we saw the seeds had cracked open and you could see a little bit of radicle. As we started giving a little bit of like drought conditions, what we saw was that the wild type and TT2 was lagging behind this BZIP and SOM lines. As we kept increasing, what we saw at 25% PEG, BZIP67 was the only one which had higher percent germination, and the other lines did not even pop open. Suggesting and kind of confirming the fact what Matthew saw, that transparent tester lines can be susceptible to a drought condition and they may not progress further. At the same time, wild type does not germinate well at a reduced water potential condition. So this is kind of a summary of these four figures where you see all the lines germinate at about 100%, but as you start increasing the PEG concentration, you start seeing that SOM and BZIP at least perform better than the wild type and TT2 at 20%, but at 25% only ZIP67 is the one which has some level of germination. But at 25%, even though they germinate, they do not transition at that high concentration of PEG. But when we transfer those seeds onto a bag with water in them, they do fine, just fine. So that's one way that we think we might be able to solve, reduce, have lines that can germinate under reduced water potential. The other thing that we wanted to test was, can we have germination at a greater depth? Because one of the things is like we are concerned about fall rain events, and if we don't have enough moisture on the top, and most of our planting methods have been either shallow planting or broadcasting, and during falls, then the drier falls, we could have issues with germination. So to test if any of these three lines that we think are good candidates for germination, we planted using this push planter from Don Wellickson at 3 different depths at 0.25 inch, 0.5 inch, and 1 inch. And we did this in 4 replicates. It was randomized within each block. And what we saw, one may ask why we don't have 100% germination here or here. That was because after we planted, we didn't get enough rain for almost 10 days. So it was kind of a dry condition. But even in that condition, we saw like about 60% germination in wild type and better germination in the transparent testa and the SOM line, which is supposed to germinate independent of the light conditions. At 0.5 inch, we saw better germination. This could be simply due to that it could capture some moisture that was there at 0.5 inch and increase the germination percentage. But then when we buried the seed even deeper, what we saw, this SOM line that just outperformed everything and the plant looked robust. So we think we've been able to solve 2 of the 3 major issues, that understanding, can we have a line that could germinate at reduced water potential? Can we have a line that can be buried? So we think we do have those 2 lines, but we still don't know how the spring temperature would, what the environment and genotype interaction would be for the first seed dormancy or the secondary dormancy in Pennycress seed. So if you have any questions here, otherwise I'll move on to the next topic.
I have a question. I'll wait till way at the end. That's fine.
Okay. As you can see that we've been trying to solve several different traits in Pennycress, and how do we keep this program going and keep this program robust as it has been. And one of the things is that we are trying to continuously develop mutant resource that would help us solve these ongoing issues or any other issues that may come up. And we are using chemical mutagenesis approach in combination with some new sequencing technologies that would make the utility of this resource even more robust. But before I get into that mutant resource, I just want to kind of give a little bit of history about what went on initially. So when the mutant resource was being developed in 2013, 2014, we were seeing David was seeing quite a few segregations for phenotypes, and they were able to validate genes. And when I got into the program, we started sequencing a bunch of lines, mutant lines of interest, and we were able to create mutation library. And that was very encouraging because that was becoming a common practice in most of the crops to build a mutation library for a species and then try to find solutions for traits or identify genes that are controlling specific traits of interest. And using that resource, we were able to kind of show that we could use a heterozygous mutation in genes like MAX3 and kind of do a co-segregation analysis and find a plant that is a dwarf and bushy. Or we could use allelic series, like 3 different plants containing a mutation in this gene that had this glossy phenotype. And using that approach, we were already having about 14,000 genes with one or more mutations in Pennycress that would be useful for developing traits or characterizing traits in Pennycress. But then we ran into an issue when we sequenced enough by the end of 2018. We saw many lines had millions of mutations, which is unexpected. When you do mutagenesis, you're expecting anywhere between 8 to 15 mutations per MB. But we were seeing a very high mutation rate in our population. And then, so I was kind of puzzled, and I started thinking, can I do pairwise comparison among these lines? And what I determined in that process was there was at least 7 different backgrounds that were as a part of the first mutagenesis treatment. And that was a big problem. And the second thing was we also had a reference genome that represented 70% of the genome, but possibly there were regions of collapse that we didn't know, and probably getting heterozygous call resulting in these hundreds of mutations as mapping with the mapping issues. And the other major problem that we had was, with the EMS, you create only C to T or G to E mutation, but that's not true. You can create other types of mutations and also some other types of indels, insertions and deletions. And because of these complexity of heterogeneous population, we had to throw out any other nature of mutations that was not C to T or G to A. And that is not helpful because you might have a line of interest, but you may be carrying mutations that you don't know, but they have a different feature. And it might be inhibiting your progress for characterizing that particular trait or that particular line. So the first step towards cleaning this resource started, even though we realized this later, but Kevin Don had already started selecting a single plant in 2014 from MN106 source and propagated that plant for several generations. And then we kept propagating, and we called it as MN106 ref. And now this seed source is available at University of Minnesota, Arabidopsis Research Center. And more importantly, this is the resource that seed source we are using for developing the version 2 genome and mutant gene index for Pennycress. Like I mentioned, before we could overcome those issues, we needed to have a pure line. So now we had a pure line by 2019. Then we needed to solve the issue of version 2, the genome quality. What we've been able to do is we've been able to use some of the new technologies, long reads, to assemble the genome in a much more contiguous manner. Like we have contigs that are as big as 64 MB. And then we have 70% of the, almost 90% of the genes genome assembled in 20 pieces at the moment, or 20 scaffolds. So we have solved these two issues, and using these two resources, now I'm going to talk a little bit about what we've done over last year or so, and how we are developing this new mutant resource and how that's being used and will be useful. So to develop the mutant resource, we started by treating 50 grams of 106 ref seeds that were produced using single seed descent and treated with 0.2 EMS. And then we broadcasted these seeds in the field. To ensure that we had good establishment, David had a very nice idea of treating these seeds with GA before we planted them out in the field. And because with the EMS treatment, usually the germination rates are very bad. But because of that GA treatment, we think we were able to get a very nice establishment. And that's the same plot that we saw in the spring. And what we were looking for is, do we see these sectors in our plots to confirm that we have successful mutagenesis in our population. And at the end of the season, we've harvested over 1,300 individual plants from these plots. We've also collected 270 pools of 10 plants from these plots that could be used for forward screens. In addition, a large representative. So once we were done harvesting 1,300 individual plants and 270 pools of 10 plants, we had these huge piles, and David had this, again, a nice idea of just running those through a trasher to just collect super pools of the seeds that could be used for various screens in the future. So we have now the seed source. How do we develop this mutant gene index? Basically, what we do, we grow these plants on a filter paper in a Ziploc bag. Once they germinate, like the radical is popped open, we transfer them to the cold for 21 days and then bring them out in these containers and then select a plant from that particular population and sequence it. In parallel, what we also do is we grow these seeds out in the field so that we can have enough seeds for that particular population which is being sequenced. So that allows us to kind of have seeds for the lines that we sequence, but also kind of look for phenotypic variation both in the field conditions and in the chambers. Like for example, we can confirm that the mutagenesis was occurred based on the phenotypes we saw, like this compact flower, big leaves, almost like a gigantea type phenotype. Or we had male sterile phenotypes that we observed, or we had albino seedlings. So this confirmed that our mutagenesis had occurred successfully, and we could use this normal process to develop mutant gene index in Pennycress. So that's with the population development. And what do we do on the data analysis side? We grow the plants, we send the DNA for core to sequence those samples. We process it through a standard pipeline where we use aligners, we use some tools for kind of correcting or removing duplicates, and then call the SNPs using GATK. And this is something that we haven't done yet, build the variant database, and that is in the books. And we are discussing how do we build that effectively. But what I want to point to you guys today is that over last 1 year, we've sequenced close to 400 lines this year. I've analyzed data for all of these lines, and what I was able to find was about 2.7 million mutations in these 400 lines, of which 88% of the mutations were SNPs and 12% mutations were insertion and deletion. As I mentioned, we had issues with the mutant gene index version 1, that we could have lost these 12% insertion deletion that is created due to EMS, or the mutations that were not C2T or G2A. And on average, we are identifying 300 genic non-synonymous mutation in each of these lines. So it's a robust resource, and here are some numbers. What I presented earlier, that we have, if you look at the ratio of mutations overall genome wide, there is about, we see that there is more bias towards genic region. We don't know why that is yet. We are investigating that further. All of this data has been analyzed in just the last month or so. So we still haven't like carefully looked into each and every detail about this, but you can see we have millions of mutations, several thousand insertions and deletions. And then more importantly, what I want to highlight is we have over 5,600 complete loss of function, which means these genes have either stop codons or frame shift. So this is a huge resource, and our goal is, this is 393, but our goal by the end of next summer is going to be sequencing 1,000 Pennycress mutant lines. And with that, we hope that we can have multiple alleles in every predicted gene of Pennycress that is at least predicted using the bioinformatics, and have that resource available for improving Pennycress, but also to understand basic biology. And one of the questions I know what may come to everyone's mind, that, well, now the technologies have improved. There is CRISPR, you can do gene editing and things like that to improve your crop. But EMS mutagenesis has some advantages that CRISPR may not be able to provide at the moment. And EMS can also provide some information that could be useful for CRISPR or gene editing type of work. For example, we can create these weak alleles that does not affect the plant fitness. Like I mentioned earlier, that we have reduced shatter lines, that do not shatter in the field conditions, and we had 3 different independent alleles. But interesting fact is that we had mutation in IND2 and IND3 that was within the conserved domain. And whenever we have had mutation in those regions, you can see that there's that layer, the cell layer is disappeared. As a result, you cannot even open those seed pods open. And that's a challenge because you could have reduced seed pod shatter, but you run the combine through and all of the pods will be intact, and that's going to add secondary layer of cleaning. The other advantage, for example, we are trying to have early flowering line in Pennycress. We have a mutation that's a single base change where we can get early flowering in the spring. But at the same time, if we did a CRISPR on this gene, we would completely lose the vernalization capacity and lose the purpose of having a winter crop for the upper Midwest. So EMS does help us create these weak alleles that would be important. And also, and CRISPR and gene editing or transformation can be are great tools, and they'll be useful only if you know what gene you want to go after. In many cases, we don't know what the gene would be responsible, what regulatory transcription factors could be involved for the traits that we are working on. For example, like we didn't know the BZIP67 or the SOM or the AOPs were going to be the targets for gene editing right away. And that's where EMS plays a huge role. The other important aspect of using EMS-based approach is it's a huge training module for training undergraduates. Like this summer alone, I had the opportunity to work with 5 brilliant undergraduates to look at different traits of interest. You can find these examples on the I Prefer website. And I'm just showing one of the ways that students can understand how to phenotype, how to genotype, and how to correlate that and establish causality between the gene and the phenotype. For example, like with this group of students, what we did, we had a dwarf Pennycress. We knew what the gene was. We grew out the plants, looked for height between the wild type, heterozygous, and the mutants. And then we were able to establish a causality between the gene and the phenotype for dwarf Pennycress. At the same time, students got the opportunity to kind of dissect where that height difference is coming between the mutant and the wild type. Is it from the first branch to the top, or is it from the base to the first branch? And that's a good phenotyping experience that student could get. But also this tool is very important module for students to learn how we can go from a trait to a gene or a gene to a trait. And there are several disadvantages too. I mean, like, you know, when you create mutagenesis, it's not targeted like CRISPR or other gene editing technologies, because you know the genes, the likelihood of you having background mutations is very low. But in case of EMS, as I said, we can create 300 non-synonymous mutations. And if you have a mutation in your gene of interest, but you have a mutation that has a much stronger expression, then you won't be able to characterize or identify that trait in that given line. And then if you want to introgress this into breeding lines, you need to clean these background mutations because you don't want to carry those other non-synonymous mutations that could impact the yields. For example, like I mentioned about how SOM mutant could be a good line for burying the Pennycress seeds at a greater depth. But that mutation was present in a line called A795. And that line had almost 800 mutations in 800 non-synonymous mutations in that. And 5 of them were interest to us, like TD4, reducing fiber, improving germination. PAD2, we can increase oleic acid. SOM, we could improve germination. IMD, glucosinolates, and DORF2 plant height. But then when we grew this line, initially, even though it had complete loss of function in this gene, we were not able to necessarily find this interesting pattern that we can bury these seeds under dark conditions, because there are other influencing factors and other genes that were mutated. So we had to clean it up. And the data I presented earlier about how the SOM is a good line is based on, we backcrossed it, we propagated it at least 3 generations. So that data was based on BC1F4. So that's one disadvantage. The other disadvantage we have is not only we create these random mutations, but sometimes we might have these random mutations that are linked to our trait of interest. For example, we had this mutation in E1396, that line we called nutty. It had mutations in gene called MAX3, which gives it a dwarf phenotype and also a bushy phenotype that may impact the plant performance. And we were able to get rid of this. But what we found after the genome was improved, and when we started looking at, this is the mutation of our interest, which causes low glucosinolate, and we started looking at flanking regions, what is present. And we found that there was a mutation that was linked with this gene AOP that could be affecting the plant development and giving this bad phenotype that we are seeing, making it look like it could be the low glucosinolate that's causing the phenotype, but it could be due to this flanking mutations. And David has really been able to kind of walk this back and see and identified a couple of lines that do not have a mutation in this gene. And also like the flanking markers are wild type, and this is a heterozygote. So even if this is responsible, we can get rid of this mutation in the next generation. So we'll have to clean this, but there are some disadvantages when it comes to EMS type of mutagenesis. This is a huge resource, and this slide will tell you why, because not only this is helping us solve some of the issues that is related to Pennycress, but it's also serving as a tool for biologists and other researchers across the country who are getting interested in learning about Pennycress and solving issues that are there in Pennycress, or they just want to use Pennycress as a supplemental model for their system. And these are the traits that I've mentioned on the left side, but like you can see that already we've distributed these seeds to several researchers on campus, to the I Prefer team, the newly funded DOE grant. We are in the process to submit those. And more recently, with the help of John Sedbrook at Illinois State, we talked to Arabidopsis Research Center and Nottingham Seed Stock Center. They are excited to host this resource for public research with some MTAs in place in the near future. So having said that, I'm just going to kind of make some few announcements or the exciting news that we had this year. One of them was this DOE iPrep grant that was led by John Sedbrook from Illinois State. This is focused towards understanding Pennycress adaptation and trying to improve abiotic stress rates in Pennycress. And then at the same time, to support this grant and also to support the gene index and other efforts, I led the project proposal for obtaining the sequencing support from JGI, where we are going to have a support to sequence about 1,000 Pennycress samples that would include 500 mutants and 500 natural accessions that is being provided by all the team that I showed in the previous slide. And then David was able to kind of obtain a grant through PPIC to understand flavor and protein extractability and how we solve that through genetics and using this mutant resource. And we also had our first Pennycress patent that was approved, that was granted this year. And more recently, some of you have got that email that it was selected to be the inventor of the year by the Bar Association of Metropolitan St. Louis. So with that, I'll just summarize that we've collected thousands of individual M2 plants. We are going to sequence 1,000 mutant lines, 500 natural accessions, and create this huge resource for the community who's interested in Pennycress improvement as a crop or using Pennycress as a model system in the near future. We've identified and validated several phenotypes for Pennycress. We are making a good progress on stacking these traits that are being identified, although this could be done much faster using CRISPR, but at the moment we are using the traditional route. With that, I'll take any questions you may have. Thank you.
Thank you, Matthew and Ratan. So it's open for questions. I apologize for the glitch in the audio. We'll have to figure out whether it's in-house or out. Any comments or questions for Ratan?
Yeah, Ratan, very nice job. You guys are doing wonderful work up there, and I think, well, you all know, and you know, just really appreciate you as colleagues. I couldn't ask for better colleagues, and what we're doing together is so complementary and it's been working very well. And looking forward to working with you for years to come here, along with the CoverCress folks who are joining here as well. So I'm really pretty interested about the BZIP 67 and SOM mutants and the prospects of those. So can you say a little bit more about those? You said that it took a bit to clean up the background of the SOM. Did you say, are those knockouts or partial loss of function? I think.
Well, so BZIP1 is a partial loss of function. SOM1 is a complete knockout.
And then, so when you did the PEG analysis, was that in complete darkness or was that under light conditions?
Under light conditions.
And so how, could you say a little bit more about the PEG assay and how good of a proxy you think that is for low water, like drought conditions?
Well, we haven't tested anything in the field yet, and that's something we're going to do next fall, this, you know, but I think it's going to be comparable. Like this year when we did even that trial at different depths, we didn't have good rain. And even then, like BZIP 67 did outperform 106 in terms of even at the reduced moisture content. But we think that it's causative, but and we do feel that our assay is going to repeat in the field conditions.
I'll say too that PEG has been traditionally used as a way to reduce water potential and look at seed germination. So it's been around for a long time.
Do you have an idea what 25% PEG, what the equivalent would that be for like a moisture content in soil, for example, or duration?
It's complicated. Because one of the things that goes on with PEG is that every PEG solution that you buy off the shelf is a little bit different. It depends on how long the polymer is. And the length of the polymer influences the degree of reduction in water potential that you achieve. And so what we really would need to do is to physically measure that water potential, and we haven't done that.
So I can imagine you can't just put a moisture probe in your PEG solution, can you?
Perhaps we haven't looked into it, but just for starters, and I think what most people do, we just did a range to see look for differences.
Mm-hmm.
Can I also ask a question?
Sure.
Well, first of all, just like John, want to sort of commend both Ratan and you, Matt, for doing a great job covering a lot of ground, and also say that we feel really blessed to be working with University of Minnesota team and all other iPrefer colleagues. I mean, without it, the commercialization of Pennycress would be in much more distant future. What caught my attention is, and we kind of discovered that as well when we were, looking for certain mutants as well, that Minnesota 106 original line seems to be heterogenic, and we actually have not seen much of that heterogeneity in some of our accessions that have been sequenced. I was wondering if you can speculate on the source of that heterogeneity and maybe share with us your thoughts about natural outcrossing rate that exists for Pennycress. We have some numbers in our minds, but want to make sure that we kind of all aligned what is maybe going on there.
Yeah, I want to say something about that. Kevin and I went around about this a little bit when we first published that draft 2 genome. And if you look at that, or excuse me, that draft 1 paper, and if you look at the draft 1 paper, what you'll see is even there, we knew that the line was actually 2 different lines that we called A and B. So early on, we knew that that 106 was mixed. And then what Ratan has found is, well, it's even mixed more than we thought. And so I think this really goes back to Don. Don, I'm going to blame it on you.
I take full.
I was just about to say that.
Full credit for genetic diversity.
So there's that.
So I'm still not clear. So why, for example, Minnesota 106 was picked? Is it just random chance? Was something, it was, it just told John, pick me, and total Dawn, grab me from the field, or, there was something special about it. And also, was the source of that heterogeneity biologically?
Well, my guess is that Don stopped along the ditch somewhere to do whatever, and that's where he found it. So Don, you can know.
Yeah, 106 is part of about 30 selections that I made in Minnesota. We grew them out, and there was some uniformity in terms of growth of the ones that we selected from Minnesota, but 106, was probably, we just considered it to be one of the best seed producers. Good standability, a nice robust plant.
Yeah.
It was in fact picked up. That one was picked up along the roadside on a field margin, right?
Yeah.
You know how it is. As you go out and you look at your phenotyping plants, right, as you go across the landscape, and that was one that caught my attention. But others similar to that, were also collected, and they were growing out, and 106 kind of became the surrogate. But the mistake, as I understand it, was that we did not do a single seed descent selection as we went into the program.
Yeah, I can add to that.
We
too.
did. It just only started in 2012.
Yeah, but I want to say one other thing too about just Pennycress in general, in the way that things were being handled early on. I think we're better now. But one of the things is that we were taking the assumption that when you harvest the seed with the harvester we were using, that we're getting a complete clean out in between picks. And at the time when I was doing a harvest of some yellow seeded and regular wild type seeded stuff, it became very clear that the clean outs were not complete. And so a lot of our stuff, I think, has suffered from that. But I think we're much better now after realizing that.
Yeah, definitely the seed source was the issue, but also something that we are still trying to understand with natural accession. There is
Kevin, did you want to say something? Kevin's online.
I very distinctly remember the day in probably 2011 that David and I were isolating the nuclei from the 7 plants that made version 1, and us vigorously debating, well, should we pick only one of these plants for the nuclei? No, we need a bunch of tissue, so we put 7 together.
So I wasn't the problem.
But you gave us the seed, Don.
Okay, I'll take all the heat. Thanks.
Gotcha. But what overall, I want to sort of hear people's speculations about potential natural rate of outcrossing that's going on. What do you guys think is going on in nature?
well, go ahead, David.
Yeah, I was going to say one of the things that we tested right away. So Kevin found these 2 different lines on his sequence, and we tested what looked to be two separate lines that were likely grown together. And I think we tested about 10 loci or more. And all those were all those were homozygous for one line or the other, suggesting that even though these two lines were grown together, there wasn't an enormous amount of outcrossing, although we all know that there's some outcrossing.
Ten loci is not a whole lot, I think, to.
No, absolutely not. But it says that at least it's not rampant.
Right.
Yeah, Tim, but also like one thing that we are investigating whether there is some level of heterozygosity among these natural accessions, because we do see that even after single seed descent of these lines, we are seeing some level of heterozygosity in Pennycress lines. Why that is, we don't have an answer yet. Like on Catherine's GBS data, we had a lot of heterozygous calls. In the natural population, what part of the genome that's coming from, we don't have an answer for that.
Okay. Well, thanks, Ratan, and everyone for the answers.
Ratan, are you guys picking a single individual plant when you're doing what you're going to do 300 or 400 of the
Yes.
wild accessions?
Yeah, so that's the plan moving forward to avoid these issues with the heterozygosity or things like that. We'll just grow a bunch of plants, but we'll just select one single plant and sequence that and propagate those seeds instead.
Any other comments, questions for this team?
Hey, Ratan, this is Russ. I have a question. So on your, I think what you were showing was percent germination for seed that you had at various depths out in the field.
Right.
Was that, were you digging up the seed and looking at germination of that seed, or was?
No, so we counted 100 seeds for those each plot before planting. And then after like 7 days, I started going out every other day and count the number of plants that popped out in that particular plot.
Oh, so it was emergence.
Yeah.
Actually. Okay. Okay. Yeah, I was just curious about that. That's pretty interesting that it was emerging from, you know, relatively deep compared to what a lot of times we actually planted at out in the field.
Yeah, so this line seems to be doing well even at the low light conditions. So hopefully, and we are in the process of combining that with other lines like TT2 and VZ67 to see could we have lines that could not only germinate at the higher depth, but also with the reduced water potential.
Thanks.
Ratan, do you have an idea what the depth limit may be? So you want an inch? Did you try deeper than that even?
We haven't tried that, but I can tell you one thing that we've been working on this depth, and there's been probably 2 undergraduates that worked on it for last 2 years. But when you do these depth studies in the greenhouse or in the lab conditions, somehow those, even if you bury them at 1.5 inch or 2 inch, somehow it'll germinate over time, because those somehow that environmental conditions ends up being optimal in the lab situation. But out in the field, I think 1 inch showed that real difference. We had only 50% germination in the wild type or 40% germination. I don't think we can go deeper than that, because one of the things that we have to consider for Pennycress is that Pennycress does have an elongated hypocotyl, and it would not have enough strength to push itself out from such a higher depth.
So maybe with a transparent tested mutant, you have a higher energy content, right? So higher oil content, maybe they'll give you a little bit more, but anyway, just curious.
Yeah.
John, do the transparent tested mutants have like more, like one seed to one seed, more oil, or is it just a higher percentage oil?
More total oil per seed. So for our TT8, we see about 12% higher total oil. compared to wild type. So if that give you 12% more, well, I don't know what the energy conversion would be for a hypocotyl length, but
Yeah, I think what he's getting at is just the absolute number of mgs of oil in a seed, not the
Right.
percent.
Yeah.
Do you have a number for that, John?
that's a good question. I have to look back at the data.
Okay.
I'm also curious, is the winter type or spring type more prevalent in Minnesota?
Winter type.
Any particular reason? Is it because of the climate or?
Oh, you mean naturally?
Yeah, naturally, yeah.
Catherine might be.
So most of the collections from southern Minnesota tend to be winter types, while most of the more northern collections and almost all of the Canadian collections are spring types.
And that's indicative of winter survival or something else?
Probably some winter survival, but also, you know, time to complete life cycle. Just, I think spring types tend to be more productive in those environments. So think about how we grow spring wheat here versus winter wheat south of I-90. So it's a very similar cutoff.
Okay, thank you, Catherine. That was interesting.
Any other comments or questions for the team? Anything that the team wants to add based on the conversation and the interaction?
I'd just like to thank everybody for showing up this week and look forward to, we do this every week, so you can come any week you like.